On behalf of our Dean, faculties, School of Allied Health Sciences, Vinayaga Missions Research Foundation, Salem, I would like to welcome you all for this International E-Conference on Basic Medical Sciences, Day 2. The prayer offered to God in the beginning of a day is the key that unlocks the door of the day. With this Medical Sciences, New Delhi, in the year 2012 to 2014, he was postdoc to Institute of Psychiatry, Munich, Germany. In the year 2014 to 2015, he was a postdoc researcher, grade 10, Institute for Integrative Sleep Medicine, Tsukuba, Japan. In the year 2017 till now, he is a postdoc researcher, grade 11, in the International Institute for Integrative Sleep Medicine, Tsukuba, Japan. He has done many projects. In the year 2002 to 2004, effects of picrotoxin injections into pedunculopontine tegmental nucleus on EEG power spectra. In the year 2004 to 2010, role of central warm sensitive receptors in sleep and thermoregulation. Almost his entire research is concerned with sleep, EEG, and neurons. Right now, he is working in role of adult-born neurons in memory consolidation during sleep. He has gone for many invited talks. He has a quite lot of publications. With this brief introduction, I would like to welcome Dr. Deependra Kumar to begin the first session of the day. Sir, Dr.
Yeah,
Deependra Kumar?
yeah.
You can begin this session, sir. You can proceed, sir.
Okay. Can you see my slide? Hello.
Yes, sir. Yes, sir.
Okay. Opportunity to present my recent paper in this so many audience students, and I'm very happy for that. So as you have requested that I have to also explain in a way that our students should understand. I will go slowly and try to explain.
Yes, sure, sir.
Usually, I prefer to take question in between the presentation, but I do not know whether that format is allowed here. But feel free if somebody has some question in between.
Yes, sir. Yes, sir.
So we know that memory consolidate during sleep, and but where memory consolidate do know. Recently, we have discovered a neural circuit in the brain that participate into the memory consolidation process. Dr. Priya, do you want me to show the video of mine, or it's okay?
Sorry, sir?
Do you want me to show my video also, or it's okay?
Yeah. Yes, sir. If you want, you can show the video also, sir.
Okay. Yeah, sorry, it seems little difficult right now, so I will proceed with the presentation.
Okay, sir.
Um, as we know that our brain encode memory when we are awake. For example, in the figure, you can see that when we are awake, the sensory information coming from our eyes, from nose, from ear, all are sampled and reach to a particular brain area, which I will discuss in next slide. And this all information is a kind of recorded for a simple presentation. Here I am showing the example of disk where we can store information, large amount of information. Similarly, in our brain, there are billions of neurons, trillions of neurons, and particularly the memory encoding area, which is a hippocampus. I will show in next slide, where all the information during the whatever we are encoding during the wake period is stored temporarily, and this all information is replayed when we are asleep. This this events whatever we are encoding during wake is replayed either in the gap of 24 hours, which is called day residue effect, or it can replay within a week, which is called dream lag effect. So this memory replay during the sleep is a very essential process for memory consolidation. The several sleep deprivation study has shown that if the animals or human is deprived from sleep, or even for example, dream sleep or rapid eye movement sleep, the memory get impaired. So we can identify this dream sleep uh, by by several parameters. For example, the brain waves, which is called electroencephalogram (EEG wave), and during the dream sleep, particularly these brain waves appear uh, around 6 to 9 hertz, which we call as theta waves. As we dream, our eye also show slow movement, and our muscle has less tension, which we call as muscle atonia. The sleep cycle in human and mouse is very different. Human is roughly a monocyclic. Monocyclic means they goes once in a day to sleep around 7 to 8 hours. But let's say the rodent, for example, mouse, they enter into the sleep cycle many times, even within 1 hour. They can complete several cycle of sleep wake, and they can transition their sleep wake stage from awake to non-REM, non-REM to REM, and then again REM to awake. So such transition is not there not exactly happening in the human in such a short frequency. Recent studies suggest that uh, memory consolidation takes place during the REM sleep. So in this science paper recently published, they have shown that memory consolidation takes place during the REM sleep. So in this experiment, what they did is they induced fear learning in mouse, and using a paradigm called fear conditioning paradigm, in which mice receive food shock when they are exposed to the new ambience. And after some duration of exposure, they receive food shock. So they are able to associate the context with the food shock, and they can generate that this space is dangerous or fearful. So after this paradigm, when the mouse return to the home cage and enter into the sleep, especially during the REM sleep, they silence the theta wave, which is generated particularly during the REM sleep. And what they found in the next day is usually mice remember the place and show a freezing behavior, but the silenced theta silenced mice showed no freezing behavior. This is the first study that shows that, especially without sleep deprivation, in which mouse sleep was intact, that memory consolidation occurs during the REM sleep. But question remains that where memory consolidate during REM sleep? I mean, in which neural circuit memory consolidate during REM sleep was not known. So we try to investigate into a particular neural circuit, which is called adult born neuron. Adult born neuron is now considered as a unit for memory encoding. You can correlate with the disk, a small disk in the brain. So, what is adult one neuron? Now, most of neuroscientists agree that adult brain can create new neuron in some of the brain area. So, hippocampus is one of the such area, especially in the dentist gyrus. In the dentist gyrus subgranular layer where the neural stem cells are present. And these neural stem cells proliferate and differentiate into the immature neuron. And at the age of 4 weeks, these mature neurons receive input from entorhinal cortex and send output to the CA3 area, which is required for memory formation. Several studies has suggested that these adult-born neurons is showing higher level of plasticity and excitability when they reach to the 4 week of age, and that is the required for memory, new memory formation. And also it is required for temporal tagging of the events, whatever happening in our life. This is encoded in a brain, and this adult born neuron is very suitable for that function. Now, what is temporal tagging of events means? If we have to recall two similar event which occur at very close, very far different time, it need to be encoded in separate brain cells, and in such job, this adult born neuron is very suitable, because as you have seen in previous slide that they are only able to encode at particular age of the development, and whatever events is happening during that period of time, that is encoded in those neurons. But once these neurons get aged, they are no longer able to encode new memory. So it is because of continuous generation of adult born neuron in hippocampus, the other batch of or other cohort of the new neurons are able to encode new uh, new memory, and this process continues. So we it was not known what is the function of this adult born neuron during sleep. So we decided to see the activity of these new neurons during the sleep. And for this purpose, we have expressed a fluorescence molecule called GCAM, which is sensitive to calcium. So when there is high calcium in the cells, these new neurons can show fluorescence, and that can be imaged using microscope. So in order to express this fluorescence molecule on the adult born neuron, we use two type of transgenic mice. One is GCAMP3, which can express GCAMP molecule on the all cells of the body, but the condition is they are stopped by a stop codon, which can only be active once the recombinase activity is higher in that cells. So for this purpose, we use another mouse line, which is nestin Cre ERT2 mice, in which nestin, which is highly expressed only in dividing cells, and they can also express recombinase enzyme. So using these two type of mice, we were able to express the adult GCAMP on adult born neuron, and particularly the thamoxifene is a chemical that is required to express the activity of Cre recombinase in the cells. So this way we were able to uh, express the G-camp on adult one neuron. Now, how to see the adult one neuron for this, and especially during the sleep. So for this purpose, we implanted a lens, diameter 1 millimeter, length 4 millimeter, and especially above the dentist iris, and we implanted EEG electrode to monitor the sleep behavior in the mice. So next, I'm going to show the activity of adult one neuron when they are awake, when they are in the non-REM sleep, and when there is a REM sleep, which we believe as a dream sleep in mouse too. So you can see this is a hypnogram, which suggests the red color is a REM sleep and the blue color is a non-REM sleep, and wake is a green color. And this recording we have done without inducing any learning in mouse. So this is baseline recording. The video I'm showing here, you can see right now here it is in the non-REM sleep, and you can see some cells are fluorescing the light. It's not only in non-REM sleep, but also in REM sleep you can see there is cells firing. But when mouse enter into the wake, the activity is much higher compared to the sleep, which is also reflected in the graph. You can see the green line is on the top and the red is the lowest. This means the adult one neurons are very active during the wake period, but it's less active during the sleep. And overall activity is they are not so uh, highly active cells. They fire very slowly and very sparse. So we try to understand what happens when we induce learning in the mice and monitor these cells again, and what happens if there is no learning and the learning in the mouse. So for this purpose, we induced learning in the mice in the similar fashion what I have shown before, which is a delayed shock, and the other in which the mouse received shock just immediately when they are exposed to a new environment, in which these mouse are not able to associate shock with the context. And in these two group of mice, we image the activity of adult one neurons during the sleep-wake cycle. So as expected, the delayed shock mice was able to associate context with shock. I mean, there is generation of fear in this mouse, so they froze more in the next day when we put the mouse back to the context. There was a memory, and the mouse froze more compared to the immediate shock, where there is no association of shock and context. But when we analyze the activity of adult one neuron during the sleep-wake cycle in these two group of mice, we were surprised to know that during the REM sleep, the activity of the adult one neuron, especially after the learning, is significantly decreased compared to the no learning group. Now, what it means, whether this decrease is really meaningful or not. For this purpose, we did another set of experiment, and in this experiment, after inducing a fear conditioning paradigm, in which mouse receive food shock when they exposed to the new environment, and after that when they enter into the sleep, especially during the REM sleep, we monitor the cells which are active, and for example the red color, and those which are not active, and also we track those cells when we put back mouse again in the next day test condition. So what we what we found is the REM active cells, the REM positive cells are also active during the learning. It means that the, the memory coding neuron during the learning is reactivated during the REM sleep. And this is probably first data that shows that adult born neurons is reactivated during the REM sleep. Uh, so far, I was showing the video of only adult born neuron, but what about the pre-existing neuron, like developmentally born neuron? Those neuron which are generated when in during the embryogenesis, or when, yeah, during the development. So in this experiment, we tagged the developmentally born neuron separately, not the adult born neuron, which is shown in the red color, but only the green color we imaged. And what we found is these neurons are not showing any significant change in the activity during the REM sleep. So the comparison of this developmentally born neuron and adult born neuron gave us confidence that adult born neuron is really important for uh, some function in the memory. So far the summary is adult born neurons shows a low activity during sleep compared to awake, and the active adult born neurons also reactivate during REM sleep. Their activity becomes very sparse and slow in REM sleep after post-associative learning. Post-associative learning means when they are able to associate context with shock, then only the activity becomes slow, otherwise there is no such change. And the developmentally born neurons activity did not change during the REM sleep. After this experiment, we asked a question whether this reactivation during REM sleep is essential for memory or not. For this purpose, we did optogenetics experiment. Optogenetics experiment is based on monitoring the cell activity through light. We express light sensor on the cell, which is generally halorhexin for silencing the activity of cells, and channelopsin for activating the activity of the cells. So for this purpose, we use again transgenic mice line in the similar way as you have seen before. To in order to express halorhexin to silence the activity of this adult born neuron, we use halo mice, and again the nestin created to mice to specifically express halorhexin on the adult born neuron. And also it's mediated by tamoxifen. Tamoxifen acts like a switch. Whenever we want to express, we can inject. Before that, it will not express. So this is also useful because we wanted to monitor only 4-week-old adult born neuron. So after tamoxifen injection, we wait for 4 weeks till this adult born neuron integrate into the pre-existing circuit. So in order to send light into the dentate gyrus area, we implanted optic fiber, which is a very thin optic fiber. Core is 200 micrometer. Nuclear aperture is 0.39, and we implanted in both sides of the dentate gyrus. And in order to silence adult born neuron in a particular stage of sleep, we implanted EEG electrodes and EMG electrodes. So here is the video. I'm going to show how we monitor, we silence the adult born neuron by online judging the sleep stages. So you can see in the video right now the mouse is in non-REM sleep because you can see the high delta waves is coming, and as soon as we see the theta waves, like for example right now you can see in the video in the mouse the light, orange light is on, we shine the light on the adult one neuron. So this is 10-second on and 10-second off during the REM sleep in order to protect, in order to not to kill the adult one neuron. So it's 10-second on and off. We did silencing of adult one neuron for 6 hours. Here I'm going to show the video of mouse. The left hand side mouse is the one which in which this mouse has received the light, but this mouse has no halorhexin, meaning the light cannot inhibit the adult bone neuron. And the right hand side, the mouse has halorhexin, and there was silencing during the REM sleep. So you can see the left hand side mouse remember the context as a dangerous, and they are freezing. But right hand side mouse, in which we have silenced the activity of adult bone neurons, they are they are not freezing, or they are freezing very less. So the next slide I will show the data. In this experiment, what we did is, after induction of fear in the mouse, immediately for 6 hours we silenced the activity of adult bone neurons, and the next day we assess their memory by putting the same mouse into the same context. And what we found is, the silenced one, the red color, you can see they freeze less compared to the blue one, meaning their memory is impaired. But same experiment when we did, not immediately after the fear learning, but later period, like for example 6 to 12 hour, the 6 hour silencing of adult one neuron, this effect was not there in the bottom graph, you can see. So it is very interesting, because the adult one neuron seems play important role in memory consolidation only in the initial period after the learning, not the later period. And this is also supported by others paper, that memory consolidation process occur within 6 hours after the new learning. So uh, then we were very excited, and we thought that let's activate the adult one neuron, and hopefully we will enhance the peer memory in the mice. And for this purpose we used chanropsin mice, and we activated adult one neuron during the REM sleep, but the result was unexpected. um, it was there was again impairment in memory. This part I will discuss in coming slide, that why the memory is again impaired after the activation of adult one neuron. For the control, we run the again optogenetics experiment, silencing adult one neuron during the non-REM sleep, which is actually longer part of the sleep. So here, mouse received light for the longer period of time, but still memory was intact. And this is very interesting, that it is not the amount of light, but it is the specific stage of the sleep, or a specific state of the hippocampus, when where the adult one neuron is contributing to the memory consolidation process. Not only the 4-week old adult one neuron, we try to see the other, for example, younger adult one neuron, or aged adult one neuron, like a 2-week old is younger, which is are still not able to make dendrites, or for example 10-week old, which is like very aged neurons. What we found is that only 4-week old adult one neuron is important for memory consolidation during REM sleep, not 2-week or not 10-week old. And even if we silence granular neuron, like developmentally born neurons, the memory was not affected. So it seems there is a specific function played by the adult one neuron at a particular age of the development for memory consolidation during the sleep. Now, as we know that memory replay or reactivation requires synaptic activity. So for this purpose, we we wanted to know why the memory is impaired when we silence the adult one neuron during REM sleep. So for this purpose, we did analysis of the spine of the adult one neurons, and in order to know the effect of silencing, we had to have another control group in which we have to just shine the light but not inhibit the adult bone neuron, or also we have to have one control group in which we induce only the learning but we should not do the silencing, and the another is without learning. Out of this all four group, what interesting thing we found is that when we silence the adult one neuron during the REM sleep, the spine neck length of the adult one neuron is enlarged. What does it mean? It means that when the neck length of adult one neuron is enlarged, their synaptic activity goes down. So what we understood is that because of our silencing process during REM sleep, the memory replay which is happening during the REM sleep, the information passing from one neuron to other is affected because of the enlargement of the spine neck. Now, what is the effect of silencing on the overall dentist gyrus structure? So for this purpose, what we did is after inducing fear learning in the mouse and silencing during the REM sleep, we selectively dissect out the dentist gyrus area and try to see the mRNA expression in this area. That what are the mRNA is changed because of the silencing. And what we found is there are some immediate early gene, for example, CFOS or NPS4 or EGR2. These neurons expression goes down because of the silencing. What does it mean? It means that the silencing of the adult one neuron during REM sleep inhibited the overall dentist gyrus activity, and that may affect the memory consolidation process. So summary of the optogenetic experiment is that silencing of adult one neuron, especially 4-week old adult one neuron, not 2-week old or 10-week old, has any effect, but 4-week old has an effect on memory consolidation, and that is also only during the consolidation process 0 to 6 hours. Apart from silencing, activation of adult one neurons also affect the memory consolidation process. So what we hypothesize that probably adult one neuron activity is finely tuned with the ongoing hippocampal activity or dentist gyrus activity, and because of that, either silencing or activating, which can impair their fine-tuned activity, uh, that results to the impairment in memory. So overall summary is silencing of adult one neuron during consolidation period in REM sleep impaired memory, and it is because there is enlargement of the dendritic spine. There is a decreased expression of synaptic related gene. So our ground summary is adult one neuron activity decreased during REM sleep only after the formation of an association between a context and saw. Adult one neurons that were active during learning were more likely likely to be reactivated in subsequent REM sleep during memory consolidation. Disrupting adult born neuron activity in REM sleep during memory consolidation resulted in elongation of their spine neck and memory deficit. So it seems there is a finely coordinated adult born neuron activity during REM sleep, and that is necessary for memory consolidation. Adult neuron number is significantly decreased as the Alzheimer disease progresses. So Alzheimer is classified in several clinical stages like BRCA stage 1 to 6, depending upon the level of impairment in memory. And this adult born neuron is progressively decreases, like in a stage 1 compared to stage 2 and 3. There is gradual decrease. Study, I am going to analyze this adult born neuron activity during the sleep in mice model of Alzheimer's disease. Thank you very much for the attention. I'm thankful to my mentors, Masanori Sakaguchi and Masashi Yanagisawa. I'm thankful to me. And thank you once again for giving me opportunity to present in this conference. Thanks.
Thank you, sir. Thank you, sir. It was very informative. Thank you very much.
Welcome.
Yes, sir. Thank you.
Participants, if you have,
Do you have any
you
question?
can post your
You can
question in
ask.
the chat box. Participants, if you have any queries, you can post it in the chat box.
I can answer if you have any question. If the time allow, I can answer right now if you have any question or clarification. If there is no any question, then thank you so much.
I don't think there are any queries, sir. I'm
Okay.
checking the chat box till now, getting you, but I don't find any queries till now, sir.
Oh, I
One is
received.
asked,
I
can you
received.
explain
Yeah,
the mice
somebody is asking question.
experiment again?
Can you repeat? I can go back and explain.
Yes, sir.
What is the question?
Can you explain the mice experiment again?
Okay. So okay. Uh, yeah, it is a challenging to see the neuron and neuronal activity in mice. So now we have a now the techniques are advancing. So we use especially this transgenic mice line. The transgenic mice line in which we can induce the gene of our interest. For example, here GCAM3 mice or nestin creatine2 gene. This gene we can induce in these mice. That's why we call this as transgenic mice. And in this mice, the expression of these genes is can be also controlled because we are putting here the switch that is called LOXP. Uh, sorry, what is the request coming? Okay, so the expression of gene is also control. Sorry.
You can continue, sir. You can
Okay,
continue.
okay, okay. Yeah. So not only the induction of gene in the mouse or induction of your interest of gene in the mouse, but also the expression of that gene can be controlled and also whenever you want. So this technique allowed us to express a fluorescent molecule on the adult bone neuron and also um, image during the development, like when they are 2 week old or 4 week old or 10 week old, because of the trigger we have, which we can do whenever we want, like tamoxifen, uh, which we inject intraperitoneally into the mouse, and it enter into the brain and bind to this estrogen receptor type 2, and that binding activate the Cre recombinase activity in the cells, and that enzymatic activity will cut out this stop codon, which is stopping the expression of GCAM3. And then after the stop codon is out, then GCAM3 can be expressed. And you can see here the green color cells are all uh, tagged with GCAM3. So after the confirmation, histologically, we implanted the lens just above the dentist gyrus area, not damaging the area. And also on the scalp, we implanted the electrode for monitoring EG, EMG. And once mouse recover from the surgery, we implanted the lens. We implanted mounted the microscope on the top of the head. Actual figure is of the microscope is here. The miniature microscope. Uh, it's very advanced right now to see the live neuronal activity in the active mouse. So, and once mouse enter into the sleep, we can monitor their sleep activity as well as neuronal activity. Is this sufficient? Please ask if you have any particular question.
Sir, my doubt was like during that experiment, keeping that two mice in the freezing box. So, what was that experiment exactly? Like when that adult neuronal this thing was removed. So, what exactly was the changes shown in the mice in both the sections? Like in one section, you said
Okay,
like
okay,
the
okay.
yeah, yes, sir.
Understood. So let me go to here. Here, am I
Yes, sir.
right?
This one. Yes, sir.
So generally, when mouse learn, because they are able to associate the context with the saw, they next time when we put the mouse, they freeze. As you can see in the left figure, the mouse is not moving out. So they are not moving. They are showing freezing behavior. This is a indication that mice remember this context as a dangerous. But in the right hand side, sorry, you can see this mouse is not freezing. Means this mouse is not able to remember that this is the place where he received food shock, and this indicates that their memory is impaired. Is that okay?
Actually, the both the mice, there have been silencing of neurons has been done in both the mice. Is it
No,
so, sir?
in the left hand side, the light was given, but this mouse, as you can see, it is heterozygote, means this mouse has no halorodopsin. Halorodopsin is a light sensor, so there was no silencing happen in the left hand side one. So there is no impairment of memory, but the right hand side silencing happened.
Sir, thank you, sir. I got it.
Oh, welcome.
Participants, any more queries? So one more query has come.
Yeah, can you read
Yes,
it?
sir, yes.
It flashes and disappeared. Yeah.
Does the theta activity during REM sleep assessed by scalp EEG come from hippocampus, cortex, or both?
Oh, great. Be recorded, of course, be recorded from the hippocampus as well as during from the scalp, both. I mean, if you are interested, please check this neuron paper in which you will have full information that we try to record not only from the scalp but also from the hippocampus, dentist gyrus, just above the hippocampus. Is that okay?
Good morning, sir.
Yes.
I have a doubt. Sir, why
Yeah,
was fear conditioning chosen as a learning paradigm? Why not any other behavioral study?
yeah, it's a very good question because I mean, um, it is very easy to induce fear in mouse. This fear conditioning paradigm is mostly used in the memory research because of the assessment of fear memory. We can understand when mouse is able to recall memory or not. Imagine if I give a pleasure response instead of fear response, we will not be able to know whether mouse is happy or not. Is that okay?
Sir, okay. Yes, sir, thank you.
Is there any other question?
I don't find any right now in the chat box.
Okay, okay, then thank you so
We'll
much.
end up the session, sir. Yes.
Yeah, thank you so much.
Thank you very much, sir. Thank you for accepting our invite and thank you for your lectures. It was very informative, sir.
Yeah, hope. Thank you so much. Thank
Yes.
you.
Yes, sir. Thank you very much.
Thank you.
Participants, please find the link in the chat box, feedback link. You are requested to fill the feedback and submit it. I'm right now sharing the feedback link in the chat box. Please click on the link and give your feedback. The next session will begin at 2 p.m. So I request all the participants to join the session at least 10 minutes before the beginning of the session, before 2 p.m. I repeat, the next session begins at 2 p.m. I request all the participants to join at least 10 minutes before the session.